The overall goal of this procedure is to better model resistant acute lymphoblastic leukemia, or ALL, in vitro.
This method can help answer key questions in the treatment of refractory ALL about how the bone marrow microenvironment influences ALL cell chemotherapeutic resistance.
The main advantages of this technique are that it is considerably cheaper and less time-consuming than utilizing traditional animal models to examine drug efficacy on refractory ALL cells.
Begin by seeding 5 to 20 times 10 to the 6th leukemic cells in 10 milliliters of tumor-specific culture medium onto a 100 millimeter plate of 80 to 90% confluent bone marrow stromal cells, or osteoblasts. Every 4th day, tilt the plate to the side and aspirate all but 1 milliliter of the medium, taking care not to disturb the adherent cell layer. Then, carefully add 9 milliliters of fresh leukemic cell culture medium, dropwise, into the corner of the plate along the side to ensure a minimal disruption of the adherent cells. After the 12th day of co-culture, gently pipette the culture medium up and down over the plate approximately 5 to 10 times to collect the floating leukemic cells, and transfer the cells into a 15 milliliter conical tube. Then, reseed the cells onto a new plate of 80 to 90% confluent bone marrow stromal cells, or osteoblasts, as just demonstrated. By day 4, 3